Let's look at this section now, inhibitors. So, it's the idea that we have all these different enzymes in our cells, or the bacteria has all these different enzymes in its cells, and we don't need every one of those enzymes trying to function at the same time. So there has to be control mechanisms. One of these control mechanisms is inhibitors. But one of the things that you notice right here, first thing they talk about is the idea of inhibitors as poisons. So certain things like cyanide, arsenic, mercury will combine with enzymes permanently and prevent the functioning of those enzymes. And if those enzymes aren't functioning, then the cell dies. So these enzymes can be classified. Well, let me go back to that for just a second. Now, that is talking about a situation where the enzyme will bond to the, where the inhibitor will bond to the enzyme permanently. If the inhibitor, if I can get all this straight, if the inhibitor will bond temporarily to the enzyme, block its activity, but then be released from the enzyme, and the enzyme goes back to its normal shape and function, then that inhibitor acts as a regulator. It regulates the activity of the enzyme. So basically, just depending on how tightly the inhibitor bonds, it can be a regulator or it can be a poison. The inhibitors can be classified as competitive or non-competitive based on what part of the enzyme they bond with. Let's go down here and look at a diagram for that. That'll be easier. Okay, so right there. So you notice you got the in A there on the left, you have the active site, and you have the substrate, and you have the matching shape of the active site and the substrate. Then in B, you have an inhibitor that fits into the substrate. So that inhibitor is competing with the substrate for that active site. So if the inhibitor fits in the substrate, in the active site, it is a competitive inhibitor. Now, in C, you notice you have another site. So any other site on that enzyme that affects the function of the enzyme other than the active site is called the allosteric site. And so if you have a substance that fits into that allosteric site and changes the function of the enzyme, which you notice in diagram C, it changes the shape of the active site, that is called a non-competitive inhibitor. It is not competing with the substrate for the active site. Okay, so competitive inhibitors and non-competitive. Okay, and they give a couple of examples here of competitive inhibitors that are used as antibacterial drugs. And so go down here and look at this. Basically, the drug right there, sulfanilamide, will inhibit an enzyme that its normal substrate is PABA. I'm not even going to begin to it. Well, para-amino-benzoic acid. So if we go down here and look at the two, you notice they both have that ring structure with the amino group there at the bottom of it. So they're going to have similar structures. So the sulfanilamide can compete. It's got a similar enough structure that it can compete with the PABA to get into this enzyme on the bacteria. And if the sulfanilamide is in there instead of PABA, well, notice what happens. The bacteria cannot make folic acid, and that's a vitamin that it needs as a coenzyme. Okay? and then the bacteria dies. If we go on down here and look at this, humans do not use PABA to make their folic acid. We have to obtain folic acid from other organisms. So we can take sulfanilamide and it will kill the bacteria, but it doesn't harm the human cells. And then here it discusses the non-competitive inhibitors and the allosteric site that we looked at in the diagram above. So let's go on down here. And again, let's see right there. It talks about the idea of this being either reversible or irreversible. In other words, if it's reversible, it can act as a regulator. If it's irreversible, it is going to be a poison. That is, if the inhibitor is reversible or irreversible. And there's an example there with cyanide and so forth. Okay. Feedback inhibition. Okay, now then, feedback inhibition. This is a really neat system. Notice what it says. Non-competitive or allosteric inhibitors play a role in a type of biochemical control called feedback inhibition or end product inhibition. So let's just go on down here, and what this does, this keeps a cell from making more of a substance than is needed, kind of acts as an on-off switch. So let's go down to the diagram right here. Real neat system. So notice what we have. On the left, we have a metabolic pathway. So you have a substrate, feeds into an enzyme. You get an end product, which they term as intermediate A. It feeds into another enzyme, enzyme 2. You get a different end product, intermediate B, which feeds into a different enzyme, enzyme 3. Now, the end product from that whole metabolic pathway can come back around and bond to that first enzyme and act as an inhibitor. In that case, a non-competitive inhibitor, since it's bonding to an allosteric site. Okay. Almost everything that happens in a living organism is controlled through a metabolic pathway, not just one sudden reaction, but a whole series of small reactions. And almost all metabolic pathways are controlled by this, what's called feedback inhibition, where the end product acts as its own inhibitor. So notice the beauty of this system. If the end product is being used up as fast as it's manufactured, then the end product doesn't accumulate, and it doesn't accumulate to act as an inhibitor. The pathway stays open, it keeps manufacturing more of the end product. But if the end product is not being used up, then that end product accumulates, and it can come back around and bond to that enzyme and block that pathway and shut down further production. That way the cell doesn't keep manufacturing more of an end product that it doesn't really need at that point in time. So, really neat system. Almost all activities that occur in whether it's a human, a plant, or a bacterium are occur by means of metabolic pathways, and almost all metabolic pathways are controlled by this feedback inhibition. Real neat system. Normally, it's going to be the first enzyme in the pathway that is shut down by this process. Because otherwise, what's going to happen is one of those intermediates is going to begin to accumulate. And sometimes that can actually cause, if you have an intermediate compound accumulating, it can actually cause damage to the cell. Okay? So, and then right there it discusses that in your textbook. Okay, so let's go on down here and look at this. Now, right here, we have another group of compounds here called ribozymes. So remember, enzymes are proteins that speed up chemical reactions without being used up in the process. Okay, in the early 80s, they discovered that certain RNA compounds could do similar type of activities, and those were termed as ribozymes. But if you read on down through this, what you find is that ribozymes are very limited in the type of activities they carry out. So basically, notice right there, ribozymes are going to cut and splice RNA and are involved in protein synthesis at the ribosomes. So pretty limited in what they will do. Okay, so that gets us to the end of that section. So let's shut down for that one and pick up with the next video. Okay.